Testing, testing. Okay. The plan for today is to start the next topic, which is the viruses. A couple of announcements first. Chapter 2 has been graded. Scores have been posted. Scantron images are not yet available. They'll be posted and uploaded on your to your Dropboxes once we get those images back. So if there are any questions about the score that appears in Tripoli, if you think there's any problem with the score, please wait until you have an opportunity to look at the image of your Scantron and compare it with the answer key before you contact Will Petrie. So Will is the TA who's in charge of scores that are posted on Tripoli for the test. So he's the person that you should contact, but only after the Scantron images are available, please. So the viruses are covered in Chapter 36. There is one section you're not responsible for, and that's the final one, which is the key lineages. That goes into more detail with the different groups of viruses than you will be responsible for. So that is not part of your required reading. So that's been made explicit. I think that's it for announcements. Okay, let's dive on in. But first, let's go ahead and start with connecting up new information with information you've already learned in the quarter. Of course, we're now starting the final third of the class. And you've learned about the domains. I'm interested in having you consider which domain or which domain or virus is assigned. Eukarya, archaea, or bacteria, which of course are the three domains you've already learned about. D, a newly identified and characterized domain that has not yet been named, or E, they're not assigned to a domain. If you're not quite sure, good chance to chat with your neighbors. Click in what you think the best answer is, and I'll check back in with you in about 20 seconds or so. Okay, you all had enough time? Click in. Okay, I'll go ahead and stop this then. Ready? Oh, good. So, approximately half of you got this right. Yes, absolutely, they're not assigned to a domain. Why is that? Viruses are not considered organisms. They are not considered alive. They're not made of cells. As you remember, at the very beginning of the quarter, we considered what are the attributes of an organism or a living organism, and that was one of the big ones was living organisms are made of cells, and viruses are in fact not made of cells, so that's why they're not assigned to a domain. The domains are strictly a way of classifying living organisms. So okay, so they're not living. So what is some important and interesting information to learn about viruses then? Well, let's start by considering this picture here. This is not from your textbook. This is a picture of a bacterium, which is sort of this purplish rectangular object here. And it, in fact, is a species E. coli. This may be familiar to you. This is the one that's in our digestive tracts along with a bunch of other microbes, and it is infected with viruses. If you're familiar with what that looks like, then you might even recognize it. These are the viruses right here sticking out. They're false colored, sort of this brownish color here as they're protruding, sticking out from the surface of the bacterium here. And so one of the first things to consider is, in fact, that the viruses are extremely small. And of course, one of the ways in which it's hard to get a feel for how small something is because when we look at pictures in a book, of course, they're greatly magnified so that we can actually see them. And because they've been magnified, they are no longer small, they're suddenly big. So we're going to go ahead and make a little field trip to a website to check out how big viruses are. They're obviously smaller than bacteria, for one. Let's see. So we're going to go visit something called Cells Alive. And let me make it a little darker in here so that you can see this better, perhaps. Here we go. Oh, maybe that's too dark. So in the image in front of you right now, you can see something familiar. So this is a way then of starting with something that you are a size to which you can relate. So here's some fingers holding a pin. Okay, the actual size scale is shown here. So this bar from here to here is about 20 millimeters, or 20 thousandths of a meter. Now, as we zoom in and increase the magnification, here we go, we're zooming in, and we can look at the tip of the pin here, the head of the pin. Once we zoom in enough, we can actually see something else appear here. There's something balanced on top of the pin, and the various things that are that will appear here are all listed here on the right, and as you mouse over, it shows up. So in fact, the first thing here is a human hair, and we can see that a bit better as we zoom in. So here again is the head of the pin. Here's a human hair, something that we're familiar intuitively with the size of that. And as we zoom in, we can start seeing some things that were not apparent before. This little pink creature right here, as we zoom in, you can see that it's something with jointed appendages. So you might not be surprised to learn that this is in fact a dust mite. As I mouse over, you can see that it appears in this otherwise blank area right here. So a dust mite is an example of a member of what? One of the phyla you just learned about, the arthropods, right? The exoskeleton-covered organisms with jointed appendages. Includes all the mites, including the dust mites. There it is. And so they're about the same size as the width of a human hair. As we zoom in more and more, we can see that there's some sort of small things that as we zoom in and in and in, we can start to finally see them. And now what are we looking at here? As we mouse over, you can see that this green spiky object is what ragweed pollen looks like. It is the size of ragweed pollen, so much, much smaller than the size of a human hair. And of course, these are the male gametophytes that we've been learning about as we went through the plants, and we're learning about how plants sexually reproduce. Next to the pollen grains, let's zoom in a bit more here. Next to the pollen grains are a couple of other objects, and of course this one here is colored purple just to make it stand out. It's not purple in real life. This is a lymphocyte or a white blood cell, and it's next to a red blood cell, which really is red in real life. So that gives you an idea of the size of pollen grains relative to the sizes of the blood cells that are circulating in our bloodstream. So white blood cells, red blood cells. As we zoom in a bit more here, we can see some other objects appearing. What's that pink thing? You can see that this is yeast, right? We learned about fungal reproduction in yeast, where the non-hyphal single-celled stages, and so a comparable size to our blood cells. Not surprisingly, there you can see now there are some sort of smaller objects. As we zoom in even more, we've got a new scale bar here showing this is about 2 microns, or 2 millionths of a meter now, from here to here at this level of magnification. These green things here are supposed to represent bacteria, as are the yellow things, Staphylococcus. So these are supposed to be 2 different kinds of bacteria. And as we zoom in more and more, you can see even smaller things appear. And these, in fact, we finally are starting to hit the virus stages. Ebola, which is this one here, you might even recognize it because Ebola has been on the news so much. It's got a very characteristic shape. This is a much larger virus than many of the others, and you can see that as we zoom in. Here's another completely different virus next to it. So hopefully by taking time to zoom in here, this will help reiterate the huge differences in size between viruses and celled organisms, whether even if they're single-celled, such as the bacterium in front of you right now. Let me get a little light back on you so you don't all fall asleep. So that's how big they are. So let's then take one virus and consider what it's actually made of. So let's zoom in here on our virus and consider its components. What is it made of? It's actually a fairly small number of components, which makes it fairly straightforward to learn about what are the parts of a virus. So basically we have a nucleic acid that is surrounded by a protein coat, and sometimes that in turn is further surrounded by an additional structure called an envelope. But viruses are not made of cells, so there are no cells anywhere in this in any of the viral groups. Now, if instead of looking at this close-up of this EM, we pull in now a figure from your book. So this is one panel within figure 36-5 from your textbook, which shows the same general type of virus now in diagrammatic form, and this is a so-called bacteriophage. This is a particular kind of bacteriophage. We don't really need to be concerned about which kind, but this is a term that is used throughout your chapter, so I think it's a useful term to recognize. This is a name given for a group of viruses that specifically infect bacteria, and they are many of the viruses that have been the most studied, and so there's an awful lot known about them. And they have an unusual shape, at least to the eye of somebody who's not familiar with looking at virus shapes. Here they are down here in more diagrammatic form, showing the different parts. But even though it looks like a complex structure, it is still basically a nucleic acid, right? So here it is, the genome, shown here in bright orange in this particular diagram, surrounded by a protein coat. So in this particular figure then, everything made out of protein is shown in green. It's not green in real life. That just makes it easier to see it. And this is a good time to point out that the surrounding protein that surrounds the nucleic acid has a particular name, and that is called a capsid. So a capsid is just simply the protein-based structure that surrounds the actual genome, the nucleic acids of a virus. This particular virus does not have, or group of viruses does not have an envelope, so we can't see it in this particular picture, but if it did have an envelope, what are envelopes actually made of? These are made of a membrane, so it's a lipid bilayer, comparable to what you're used to looking at cell plasma membranes, but this is not a cell, so it is not considered a cell membrane. And it also often contains proteins embedded in it, just as cell membranes have proteins embedded in a lipid bilayer. Okay, but there's no envelope in this particular viral type. But let's check out another type. This is another panel from the same figure, still figure 36-5, and this shows another kind of virus. So as already pointed out, so the text in the top half of this paragraph here is exactly the same as what was on the other slide. Here's the new information about about the nucleic acids that make up the central part of viruses, in complete contrast to what we have seen with living organisms, all of which are double-stranded DNA. Viruses can have DNA or RNA, and it can be single-stranded or double-stranded. In fact, this is a way in which viruses are identified and classified is by the nature of the nucleic acids that make up the central part of the virus, whether it's RNA, DNA, single-stranded, or double-stranded. And this particular example of a virus looks completely different than the bacteriophages we were just looking at. If you look at the name up here, it says tobacco mosaic virus, so this is in fact a virus that infects tobacco plants, among some other plants. And here in this particular diagram, again, you can see the genome middle here. It's yellow in this particular diagram, and you can see it's in a helical shape. The overall arrangement of proteins is completely different, and this is something that virologists use to identify particular virus types, is that they look different. The overall structure is completely different. So a virologist could look at that previous picture and go, this is a particular kind of bacteriophage, and similarly somebody who is familiar with this particular kind of virus can identify it as such. So the capsid here is shown in green, and so you can see it's a group of units that all are interlocked together to form this sort of cylindrical structure. So completely different in appearance. The fact that this infects tobacco plants then reminds me to remind you all that viruses can infect different kinds of living cells, whether it's plant or bacterial or animal or fungal or whatever. And let's look at one more. This is the smallpox virus. This is an example of something that actually does infect humans. This is our first example of an enveloped virus. So in addition to the genome, which is shown here in this diagram in bright orange, and the surrounding structure made of proteins, the capsid, which is shown in green in this particular figure, we also have yet another layer surrounding this virus. And in fact, you can see there are two separate envelopes here, and so you can see the little structures indicated here. These blue and yellow objects here are presumably showing molecules that are sticking out of the membrane or the envelope, probably proteins. So this is an example of what an enveloped protein looks like. So that's what viruses are actually made of. And now let's consider, how do viruses actually replicate? If they never replicated, then they would not be the serious disease-causing agents that, in fact, they are. So the first thing to consider is that viruses are obligate intracellular parasites. Now, as you read something like this, if you don't understand or recognize what the adjectives mean as you're reading the textbook or listening to a lecture, this is a good time then to stop and look up the exact meaning of these words, because it's very important for understanding the concepts that are underlying these sentences. So obligate means obliged to, obligated to, it's necessary. So that means the only way in which viruses can replicate is if they're inside other cells, right? If they're inside cells. So that's what intracellular means, right? Inside the cells. So they absolutely cannot replicate outside cells. Parasites just simply indicating that they are using up the resources provided by the host cell without providing any kind of benefit in return. So each virus type then typically has a fairly limited number or type of specific cells, host cells, that is capable of infecting. An example of this is shown in figure 36-1 from your textbook, which is in front of you right now. At least this is the first, or the anterior half, the top half of this particular diagram. So it shows a couple of different body parts in a human with examples given of some of the diseases or viruses that are that typically infect those parts. So dengue, Ebola, polio, rabies, West Nile, many of these are probably familiar names to you from the news. HIV, measles is the disease caused by this particular virus, and by the paramyxovirus. And so this should immediately alert you to the idea that the reason that this figure is drawn as it is because a given virus can't necessarily infect all the cells even within a specific organism. And in addition, types have some specificity also in terms of what kind of cell they're capable of, in terms of what kind of organism. And so, for example, some viruses can only infect one particular species of plant or animal. Others can infect a couple of different species or types of hosts. But there's a lot of variation in that. And one other point that I hope is really clear is that as you were looking at the different kinds of viruses, DNA versus RNA, or single-stranded versus double-stranded, or infecting tobacco or infecting something else, these are completely different kinds of virus. These are not different stages of the same virus. So that is those are characteristics of a viral entity which can be recognized as separate from the other viruses that have different characteristics. So if a particular virus is a double-stranded DNA virus, then it is a double-stranded DNA virus always. Something that's not always very clear. And of course, one of the reasons that we're so interested in learning about viruses is they cause a lot of diseases, and many diseases are perhaps less familiar to you that are discussed in the chapter, and other ones are familiar. Influenza flu is quite familiar to all of you. Every year we hear about the flu shots that are being produced that year. But what you might be less aware of is that the influenza flu can be a very serious disease compared to some of the other diseases that are plaguing humans in different parts of the world. This may not seem like such an important disease, but an example of how serious influenza flu can in fact be is shown by the data from the terrible flu pandemic that occurred in 1918. It is referred to as a pandemic instead of just simply an epidemic because it occurred all over the entire globe. And figure 36-2 from your textbook, which is reproduced in front of you right now, shows an example of a hospital ward during the photograph that was taken during this time. And it's hard to even imagine something that is affecting so many people globally. Keep in mind, of course, too, that in 1918 the global human population was much less than it is now, and so infecting 500 million people, which is still an extremely large number of individuals, was an even larger proportion of the total human population at that time. So this affected the entire globe, and between about 50 and 100 million people are thought to have died during this epidemic. So a terrible epidemic, which is one of the reasons why flu is in fact taken quite seriously, because we have recent evidence for the deadliness of some particular strains. So how are viruses spread? Well, I couldn't resist showing you this particular animation. So this is calculations that were done by scientists who were analyzing, somebody coughs or sneezes on an airplane, how does it actually spread? So here we have a person, we're in the inside of a plane here, and what this shows, all these little dots here, are showing that the spread that would be anticipated given the geometry of the inside of a plane and the circulation patterns that build up. And you can see that it's sort of circulating, sort of around and around. And so obviously this person didn't cover their mouth before they sneezed here, and so there's just a few more seconds here, and then I think it's going to show the areas where individuals are particularly vulnerable to catching this or being exposed then to what's being sneezed out. Oh, looks like I truncated it before that part. All right. So why am I showing you simulations of the travel of a sneeze? Because of course that is one of the ways in which some viruses can be spread, but that actually goes back to this point right here. How is a virus transmitted between different individuals? It's highly dependent on what cells on an individual are attacked by the virus in the first place. So one of the things that makes cold viruses and influenza flu viruses so incredibly contagious is where they are located. They are typically infecting cells that are associated with the respiratory system, and hence any kind of sneeze or cough then can propel the viruses out into the environment, as opposed to viruses which are not associated with respiratory systems. So some viruses are much less infective, and actually contact with blood or other bodily fluids is necessary for transmission of viruses. Completely different from viruses that cause influenza or colds. Okay, so how do viruses replicate? We'll spend some time now going through the parts of figure 36-6 from your textbook, which is in front of you right now. And before we walk through this, I want to put this in the context of being one of two major ways in which viruses are replicated. So this first category is referred to as replicative growth, or an example of a lytic cycle. And so we'll walk through this first, and all the steps are shown here. And as you walk through steps in these viral cycles, I think what really helps the understanding here is to recognize that replication means that you end up with copies being made that are exactly the same as the original viral particle. and I'll return to that point a little bit later. So here at step number 1 then, in order to get replication, given that there are obligate intracellular parasites, this means that a viral particle has to end up in the vicinity of the right kind of host cell within which it can replicate. So first the virus has to actually reach the cell, and then when it reaches the cell, the very first step is that the nucleic acids have to enter the host cell. There are a couple of different mechanisms for this. I'm going to shrink this down and move it to the side so that you can see this simultaneously in sort of more of an animated form on the right as we consider this. So I'll go ahead and start up this animation. This is supposed to represent a host cell here. Along comes the virus particle, and there we go. In this particular example then, we have the nucleic acids of this virus then entering this cell. And so this is a typical way in which bacteriophages, for example, infect bacteria. It's sort of injected directly into the cell as shown here. So that's step number 1, is that the genome has to actually enter the cell. Then what happens? Well, again, keeping in mind that we have to end up with copies of the virus. What are the parts of the virus? Well, this particular one, because it's a bacteriophage, there's no envelope. So we just have what? Nucleic acids, the viral nucleic acids, and the protein that surrounds it, which means what? That within this host cell, we have to have 2 things being made. We have to have copies of the viral genome being made in whatever form it is, whether it's DNA or RNA or double-stranded or single-stranded. We also have to make the proteins. What are these proteins that have to be made? The capsid, right, the name of the protein-based structure that surrounds the nucleic acids, and also any enzymes that the virus has to provide because it's not part of the normal host cell biochemical machinery. Let's go on to the next step here and consider what happens next. So here we go, again, here's the nucleic acids being injected into the host cell, and then what happens in many, but not all host cells, but what often happens is that the host cell DNA is degraded, and it looks like we've already had replication now of the viral genome. So we've already had replication of the viral genome. Now what? We need replication of the capsid, the proteins. And so here you see this in sort of an animated form. So these are points 2 and 3 in the static figure from your textbook. And then once these parts exist, they have to be assembled within the host cell, and then released to the outside. And that's actually where this name comes from. Lytic cycle is the same root as lyse, to break. So this is so there are a couple of different ways in which viral particles can be released to the outside, but lysing the cell is one method. And of course, if the cell is breaking open to release the viral particles, this is kills the cell, and so the cell has been damaged. Let's see, what's I want to out here. Oh, there was one thing I also wanted to say back with this point here, is that of course, depending on where the host cell is taking up residence, replicating, that's associated with the symptoms of the disease that is caused by the virus, right? You're going to have completely different symptoms if the virus is replicating in cells of the respiratory tract versus your liver cells, for example. Okay, let's head on back here. Okay, so that was then the I don't think there's anything. Yes, the next point is the so that's all I was going to say then about this particular cycle, and we'll look at another variation on this later. But now let's consider the other major category of ways in which a virus can be replicated. It has a different name because it's a different process, and so this particular process is referred to as a lysogenic cycle as opposed to a lytic cycle. It is also described as being an example of viral dormancy or viral latency. And so in this process, which is outlined in figure 3613, which is in front of you right now, Then again, we start with the very first steps of infection. We've got our viral particle. I don't remember if I pointed out an individual viral particle is called a virion. This is the name given to the viral particle when it's extracellular. It's outside the cell, so that's what's meant in your reading. So once the viral particle or virion comes in contact with the host cell and injects its genome, or its genome ends up in the host cell depending on what kind of virus it is, then the next step is where it's completely different. The next step is labeled here integration, where the viral genome is actually integrated into the host cell genome. Let's go ahead and shrink down this figure a little bit and shove it to the side so we can watch this also in an animated form. I think this will make it a little bit easier. Here we'll start with our host cell. There's a genome in there. This is supposed to represent a bacterium, which does not surprise you because it's circular here. So here comes our viral particle, and it's going to go ahead and inject its genome or nucleic acid into the host cell. Here it is, but now it is going to go ahead and become integrated into the genome. So notice that we now, that's already happened, and we now, it's color-coded so you can see what went where. And so at this point then, of course, the cell would be relatively asymptomatic because it's not doing anything to disrupt the host cell. And so what happens now is just simply any time that the host cell replicates, it replicates the integrated viral genome along with its own genome, right? So if we go to the next little tiny animation here, this is an animated form. So here we have a cell, and then when it divides and it copies the genome as a whole, well, the viral genome is being copied and copied and copied too, right? So we're still ending up with multiple copies of the viral genome, but up to this point, it's just embedded in the host genome, and so there's no, again, this is asymptomatic from the host cell point of view, and it's certainly not at any kind of infective stage, because it would have to get out and infect a new cell, right? So how does that happen? Well, that's the final step in this figure here, as labeled. At any point after integration, the virus may activate the replicative cycle. So back to the lytic cycle where the viral particles will be made and then will bust out of the cell. So let's go ahead and watch that next step here in an animated form. So here we go. It's coming out, and then it will take over the host's biochemical machinery, direct more copies of the viral genome and the proteins, and now we have more infective particles as just shown here. So the trigger for switching then from the lysogenic back into a lytic cycle then is somewhat viral dependent, but it's usually associated with some kind of stressor on the part of the host cell. So as we're considering then the details of viral replication, I think the best way to wrap your mind around this conceptually is to consider what's going on in a host cell and what is a host cell sort of set up for. And so if you look at this is not a figure from your textbook. This is just simply a conceptual diagram to encourage you to wrap your mind around this. So what do host cells do? They're double-stranded DNA for one, right? As opposed to the viruses. I've got a little list on the left. Viruses can be double-stranded DNA like the host cell within which they're replicating, but they can also be single-stranded DNA, double-stranded RNA, or single-stranded RNA. But what are host cells set up to do? Well, host cells certainly are capable of replicating double-stranded DNA, right, during cell division. They're also good at generating mRNA from DNA and also for making proteins from RNA. So that's the typical machinery that already exists in the host cell that the virus can tap into, okay? But if the virus needs the host cell to perform actions that the host cell is not already up for, then the virus needs to bring in its own enzymes to facilitate and complement what's already going on in the host cell. So this is shown diagrammatically in figure 3616, which is now in front of you, which is an overview of the different ways that a virus can replicate. And we've got several different major classes of viruses as shown here. And so if we start on the left, we've got two classes here labeled DNA viruses. So we've got double-stranded and also single-stranded DNA viruses. And then moving to the right, we've got some RNA viruses here, double-stranded RNA, single-stranded RNA, and then we've got both plus and minus. The plus and minus refer to the so-called sense of the RNA virus. And what does this mean? If we go back to thinking about what a host cell does when it makes protein, it uses single-stranded RNA to generate the protein, and so this would be considered the plus sense. So that's why we have two different kinds of single-stranded RNA. And then over here on the right, we have the so-called reverse transcribing viruses, which are somewhat different. We've got an example that is both RNA-based and DNA-based, as shown here. One of the things that makes it easier to navigate around this diagram is to recognize that red in this particular diagram stands for DNA and yellow for RNA. That makes it a little bit easier to walk through this. And down at the center bottom of this particular diagram, the scientific illustrator has chosen to put the messenger RNA making proteins as one of the items that is common to all of these different viral types. Again, the proteins that are being generated, you should be thinking to yourself, right now we're considering viral proteins. We're completely ignoring all the things that the host cell itself is doing. So viral proteins include both sort of more structural proteins, namely the capsid proteins, but also any kind of enzymes that the virus might need. So why would the virus need more enzymes? Well, consider how different the steps are for these different kinds of viral virus types, right? A virus that is double-stranded DNA should have an easier time plugging into the host cell machinery, right? After all, if we go back to the little cartoon of the host cell is already set up with double-stranded DNA, already knows how to replicate double-stranded DNA, already has all the enzymatic wherewithal to make more copies and make proteins and so on. So this particular class of DNA viruses shown over here on the far left is actually pretty straightforward, and there is no necessity for viruses of this particular kind to bring in or involve any additional viral enzymes to help the whole replicative process. But notice over here on the far right, we actually have an example of double-stranded DNA virus that is undergoing a more convoluted route for generating viral products. And so you can see we're starting with a double-stranded DNA here and then making RNA. But notice now there is something different going on. We've got DNA being made from RNA. This is really quite different than what the host cell is set up for, and this is then facilitated by or catalyzed by an enzyme that is part of is one of the proteins then that is coded for by the viral genome, reverse transcriptase. This is not something that exists in the host cell. This is something that the virus has to bring with it. So as you think about the actual steps that are involved in replication of these viruses that are involving this new process, then this absolutely requires an enzyme that the host cell does not normally have in its normal functioning replication here. Let's see, what else did I want to say about this particular part? Actually, I'm going to go ahead and stop right here. You get out a few minutes early here. Nice little break for you. So make sure you review this diagram. And on Wednesday, we'll start with a clicker exercise for considering how viruses of different kinds can plug into the host cell machinery. So I'll see you then.